I've spoken to you guys a couple times about my own personal colonies. I have talked to you a few months back about the Layens hives, those horizontal hives that we have been working on. But the other major research that I've been running this summer with Nod, so I work for Nod, they're a researcher. We are the ones that make Formic Pro. So the other research that I've been running this year is a study looking at viruses, and we've been focused on a particular version of deformed wing virus called DWV-B, and that's the one with the most direct relationship to Varroa. So there are other types of deformed wing virus. Actually, I think people have identified about 40 strains of it now. But DWV-B, which some other people have called Varroa destructor 1 virus, is known to be directly vectored by Varroa. Just to give you a bit of background why we wanted to run this research, probably most of you are aware that Varroa prefer to hang out on drone larvae and adults. So they tend to target drone cells to reproduce in, and they also target drone adults. We don't know for sure, but there is quite a bit of evidence to suggest that they have to feed on adult bees too. So when they are feeding on an adult bee, they tend to feed on drones or they tend to hang out on drones. And when they're targeting to get into the cell to actually undergo reproduction, they're targeting drones. And the reason for that is thought to be because drones spend more time under the sealed brood capping. So that's where the Varroa is protected. The bees can't get in there and remove them. And that gives them a longer opportunity to produce offspring. So drone production varies throughout the year. I think that most of us are probably aware of that. So that it ramps up in the spring when colonies are swarming and queens want to mate. So drone production increases and it kind of peaks in like late spring, maybe into the summer. And then as you get closer into the fall, the numbers start to decline. And towards late fall, eventually the colony stops producing drone brood and all of the adult drones are actually kicked out of the hive. Worker production follows a similar arc, but it's not quite as cut and dried. So we do see an increase in the production of worker brood from spring to summer. It also diminishes later in the season as they're getting ready for winter, as they're going into, in some places, a broodless state. We also, here in Ontario, even though we're quite a bit further north from Maryland, I can go into my hives basically at any time of the year and still find a little bit of brood. So I don't feel that we really have a truly broodless period, but there's certainly a reduction in brood production. So that's true of workers, definitely true of drones. So drone brood production stops, whereas worker brood production really just slows down. We also see that the incidence of overt deformed wing virus, so when we actually see clinical symptoms of deformed wing virus, we tend to see that later in the season. We tend to see that in the fall. And consequently, colonies also tend to crash in the fall because of mite and virus infestation. So what are our hypotheses for this particular study? We think it's possible that drones are actually acting as a reservoir and also a buffer potentially for the rest of the virus. So essentially the idea is this: when you've got drones in your colony, most of the virus is going to be concentrated in the drone population. That's because Varroa spend most of their time with the drone. There are other routes of transmission I should mention for viruses. So they're not only transmitted by Varroa, they can be transmitted from bee to bee as well. And there are other routes of transmission, but Varroa is the primary transmission route. So if there are drones in the colony, we think that it's possible that the viral titers, the levels of virus, are building up in this population. But to a certain extent, the drones are actually shielding the rest of the colony from the virus. So the drones are acting as a buffer. Again, I think most of you are aware that the drones don't really contribute to the functioning of the colony. So from a colony perspective, if the drones are filled with virus, they're not very healthy. That doesn't really matter for the functioning of the colony because the colony function depends largely on what the worker bees are doing. Of course, the drone's job is to mate with an unrelated queen. Whether or not that happens, it doesn't affect the performance of that particular colony. So then consequently, in the fall, when the production of drones decreases, that's when we see the increase of clinical symptoms of DWV, and we're mostly paying attention to that in the workers. And we think that that's not just because of the overall reduction in brood production and also overall shrinking of the colony size. So now you've got way more Varroa per bee, but also because their preferred host, the drones, aren't there anymore. So they're host switching. We actually think that's probably more important than the fact that the colony is just shrinking. Probably some of you have seen Randy Oliver's graphs over the season where you've got like the rise and the fall of the honeybee population, you've got the rise and the fall of the Varroa population, and those lines don't exactly line up. The peak of the honeybee population happens before the peak of the Varroa population in a naturally coexisting colony. And again, yeah, we think that what's happening, drone brood's not being produced, Varroa have to go somewhere. So now they're starting to parasitize worker brood. They don't want to be there, but that's the only choice that they have. And then all of the virus that they have that's built up in that Varroa population is getting transmitted to the worker brood. Now that actually matters, that it's actually having an effect in the colony. And so the outcome of all that, if that's true, is that the timing and the type of Varroa treatment that you use should actually impact both viral load and colony performance. So things like honey production and things like population size, in addition to if we look at the load of viruses in the colony, should be affected by how you're treating for Varroa. So we've been running this study for the last 2 years. We have funding from North Dakota Department of Agriculture. It's a collaborative study with a lab there that does all of our virus testing for us. But the first year was kind of a smaller version of it. And so I'm going to show you data just from this year because it's a more complete data set. So this is what we actually did. So that's my research apiary, or one of them. We've actually got 4. We have normally about 30 hives here, but in the study there were 24 hives. So essentially, we set up the apiary around the end of April. So we split where we needed to. We requeened as needed. We actually requeened with black Carniolans from Australia at that time. So Australia is a big source of queens for us, especially that early in the season. We can't produce them here in Canada that early. So we set up our 24 colonies and we did our first round of assessments. So we went in, we took weights for the colonies. We actually have a scale. We've modified all of our hive stands. But we've got this special scale that's actually produced by a company in Canada called Hyvetronics. It fits underneath of the colonies, and then it kind of operates like a pallet jack. You kind of like pull a handle down and it jacks up the hive. So it's actually a huge advance over how researchers have been weighing hives in the past, which is like usually picking up hives and using some kind of tripod device, which is really inconvenient, or using a suitcase scale or whatever that's not very accurate. So this is very accurate. It hooks to a smartphone. So we take colony weights. We also estimate population. These are 10 frame Langstroth singles. That's what we normally run. We estimate population by looking in the top and underneath, and then we take the average of how many seams of bees we see. So we actually run 9 frames in our boxes. So even though they're 10 frame boxes, we actually run 9 frames in them. So we've got 10 seams of bees possible. That's the maximum. So we've got a number from the top and from the bottom. We also estimate how much brood is in there. We do that in the field so that we can capture eggs and young larvae, but we also take pictures of the frames so that we can get a really good estimate of capped brood. The problem with taking pictures is that you can't really see the eggs or the young larvae in there. So we have to do those in the field. And essentially what it is, it's a method called the Liebfeld method. You look at the frame. You can do it in slightly different ways, but you can either physically overlay a grid on top of it, or you can just imagine that there's a grid there. What I do is I look at a frame and I mentally divide it into 4 quadrants, and I estimate in my head how many of those 4 quadrants are filled with whatever it is that I'm counting, in this case, brood. And then I flip the frame over and I do the same thing on the other side. And then I write down a number out of 8 for each frame. So what the area of brood is there. We also look at things like queen status and identity. So we mark all of our queens so that we know who they are and if they've undergone supersedure or whatever. We do alcohol washes. This is the thing that takes us the longest. We do operculated brood biopsies. If you see in the middle 2 pictures there, pulling out 2 frames. So we want a frame that has primarily worker brood, and we want a frame that has primarily drone brood. Sometimes we're lucky and we call them one frame wonders, and they have enough drone brood and enough worker brood that we can just pull 1 frame. But oftentimes we have to pull 2. And then we're going to uncap a random 100 cells. We're going to go in there. We've got our special magnifying field glasses here. These are dental loops is what they actually are. We look at what's in there. So we say, is it a purple-eyed pupa? Is it a pre-pupa? Is it a white-eyed pupa? Is it alive? Is it dead? And then we see if there's any Varroa in there. And then who are they? Are they adult females? Are they immatures? Are they males? Are they alive? Are they dead? This takes an incredibly long time, but I have to say after 2 years of doing this and looking in thousands and thousands of cells, I'm now much faster at it. But it still takes a very long time. When we do that, we also pull out samples that we send away, and I'll get into that in a second as to what we actually pull out for samples. We're targeting purple-eyed pupa. So we want to have pupa that are at the same developmental phase across the board. So we're going to pull out 6 of those from workers and 6 from drones, and 6 that are infested with Varroa of those, and 6 that are not infested with Varroa. And then we're going to pack those away in our -80 freezer and eventually send them off for analysis in North Dakota. We also run sticky boards. So that is the screened bottom board that people are talking about. And we have these trays underneath of them. Every 3 days, we go in. We actually use manila file folders. We coat them with Crisco. I don't know if you guys have Crisco in Australia. It's used for baking. It's like a lard replacement. It's a hydrogenated vegetable oil is what it is. I'm sure that you have something similar. We coat the manila file folders with this, and then it makes it so that if the Varroa fall through this screened bottom board onto this sticky board, we call them, they stick there. So every 3 days we go in, we pull those out and swap them out for new ones. And we then take them back to the lab and we count what's on the sticky board. And so we've got an entire season's worth of data for that. And then on top of that, we track our honey production. So how much honey we've actually pulled, so we know how productive these are. So on the right-hand side, we've got the overall timeline. Like I said, colonies were established in April. We did our first assessments at the very beginning of May. Then we had 4 treatment groups. We treated our colonies on May 16th. We had 2 strips of Formic Pro treatment group. Then we had a treatment that is legal in Canada and the US, but can't be used, unfortunately. It's not on the label in Australia or New Zealand. And this is this 1+1 treatment. So that's where you put a strip in for 10 days, and then you put it in the second strip for 10 more days. So rather than putting in 2 strips simultaneously, you do that. We get lots of questions at NOD about that treatment, and honestly, we don't have a lot of data on it. So that was one of the reasons why I wanted to run that as a treatment group. So we ran that. In addition, we also have an InstaVap. So we ran an oxalic acid vaporization treatment. We did that 3 times every 5 days. So we did it 5 days apart, 3 times in the spring. And then we had a group that wasn't treated. So in each of those groups, there were 6 colonies. So we had 24 colonies altogether. It's not a huge sample size. I would have liked to have had more. But first of all, the virus analysis is very expensive. And then second of all, the people that you see in that picture there, me and my tame beekeeper, Hannah, we are the entire research team. So there's only 2 of us. This takes us a very long time. Okay, so then we went in every month thereafter up until our last assessment was in October. I don't actually have the dates here, but it was around the middle of October was our last assessment date. Just getting ready to ship off our last virus samples to North Dakota actually for analysis. So I don't have the full viral data set. The last 3 data points haven't gone for analysis yet. Just a little bit more on that. So first of all, on the left-hand side, some of the many, many, many pictures I've taken of big families of Varroas that you can see in cells sometimes. All the white guys are immatures. There are probably a couple males in there. It's a little bit hard to tell from these pictures exactly. In the middle row, on the right-hand side, the furthest to the right looks like to me like it's a male. Those other 2 that are next to it, those are immature females at a later stage of development. And the little ones, you can't really say for sure when they're that age, but they are probably females. And then on the bottom picture, you can actually see a mature female in there. There's this story that it's always only one foundress that gets into a cell. But I can tell you that I often see it where there are 5, 7, 8 foundresses in there. For some people, it's been a puzzle how Varroa survives so well, given that they're super inbred, because the mating takes place in these capped cells, and it's basically brothers mating with sisters. So they're so inbred. And the thought was they've traded genetic diversity for the ability to remain protected within these cells. But it turns out that they haven't really. And if you look at them genetically, they're more diverse than what you would anticipate if they were super inbred that way. And I think that that's the full explanation. It's not just one foundress getting into these cells. It's multiple foundresses very often. This was just the sampling, but I already kind of told you about that. So what we sent off for viral analysis at the end of the day, from each colony, we have 36 samples that we're sending. So we're sending 6 drones with Varroa, 6 drones without Varroa, 6 workers with Varroa, 6 workers without Varroa, and those are purple-eyed pupa. And then we're also collecting 6 adult drones and 6 adult workers, and we were targeting foragers. Normally, what we would do is we would pick them from the honey super, so we were reasonably sure they were foragers. You can't always say for sure. So we sent them off to North Dakota. That's my collaborator, Zach, on the side there. He was always blinded to what we were sending him. And in addition, when we were sampling from the yard, we tried to blind ourselves as much as we possibly could. Now, because there's only 2 of us, it's a little bit hard. But I will tell you, these days are long. There's a lot of stuff to do. I can hardly remember my own name at the end of some of these days. We would be out there for like 14 hours doing these brood biopsies. We should have been reasonably blind. Preliminary results. What did we actually find so far? Here's what we see if we look at viral titers in the colonies. We did actually test for 3 strains of DWV. We'll just talk about B because, like I said, that's the one that's transmitted by virus. We looked at A, and it's completely uncorrelated with whatever was happening in the Varroa population. So I think that just adds more evidence to the fact that it's not being transmitted by Varroa, really. And then we also tested for C, which we didn't find at all. So DWV-B results were the most interesting. So here's our 4 treatments on the left-hand side. You can see when we applied the treatment. This is the adjusted titer levels in the adult worker bees, which to my mind is the most important population when you're talking about who is harboring the most virus. So before treatment, there was a certain level of virus. After treatment, the 2 formic acid treatment groups, the virus load was suppressed quite a bit. The oxalic acid, interestingly, doesn't look any different at all. And in fact, that was true May, June, and July. The viral titers in the oxalic acid treatment group don't change, which is interesting. And then in the non-treated control, they actually go down a little bit, but that's not a statistically significant difference. And then by July, they've started to really ramp up. So we can see that the titers in adult workers do change in response to Varroa treatment. I'll have more data on this because we did a fall treatment, and I'll have that captured in the next set of analyses that we have done in North Dakota. Unfortunately, I don't have that data yet. This is alcohol wash data. I apologize that the bars are a little short here. So before treatment, because it's fairly early in the season, alcohol wash percentages are pretty low. That was like the May 1st time point. They start off pretty low. This is like below treatment threshold, really, or right around treatment threshold, maybe. We applied treatment here. The 2 formic acid treatment groups go down to effectively 0. Like they're we had 1 or 2 in the whole population. And interestingly, again, oxalic acid vapor, and we didn't run that group to make oxalic acid vapor look bad. We ran it as what's called a positive control. So we wanted to see what the response was when we had another Varroa treatment, which should work, especially that early in the season. There's not a ton of brood in the Canadian colonies at that point. Be that as it may, we had an increase in our Varroa loads in both the non-treated and oxalic acid groups. And then by July, we still have like reasonably low Varroa levels in the 2 formic groups, but much higher in the oxalic acid group. One of the things that's more interesting to me was that there is no difference between the 1+1 group and the 2 strips group. That's not what I would have predicted. I would have really thought that the 1+1 group would have not done as good of a job. But I think what's happening is because it's on for longer. So there's essentially 2 treatment periods, even though you got less formic acid going into the hives at those times. So I'm going to run this again next year and see what happens with a larger sample size. I'm really curious. Now, what happened in our brood infestation levels? So if we look at the infestation levels in drone brood before treatment, it's about 20% across the board. It's actually a little bit lower in the untreated controls just randomly because we can't analyze all this data before we assign them to groups, unfortunately. So it just happened to be the case that it was a little bit lower. After treatment, though, in both the 1+1 and the 2 strip group, it really suppresses the level of drone brood infestation. Oxalic acid, a little bit, but it's actually not any different from the untreated control, which didn't change from the level that it was at in May. Interesting. By July, we start to see the 1+1 and the 2 strips groups increasing again. Oxalic acid is higher and untreated control is really taking off. So this is the level of infestation of drone broods. By July, this is like 40% of the cells that we uncapped in the untreated condition had Varroa in them. That's high. And I will tell you, those are painful brood biopsies to do. It takes forever. And then this is really the thing that matters the most, which is the worker brood infestation. We always see a little bit of worker brood infestation. So there's always like 1 or 2 cells, usually out of 100, that have a Varroa in them. So that was the levels before treatment. After treatment, similar pattern to what we saw in the drone brood. So the formic acid groups go down, oxalic acid group didn't really change, and then the untreated control is increasing. And by July, we actually see quite a big increase in the oxalic group and the untreated control group. So that's getting up to like 6 or 7% of worker brood infested. That's actually getting pretty high. That's not a desirable level. And then with respect to the original hypothesis that we had, which is that the level of drone brood in the colony should be related to the amount of viral titer in the adult workers. That's really what we're saying. We're saying that when there are lots of drones in the colony, we should expect that the adult workers should not be that infested with virus because they're being protected by the drones from that virus. But when there's not very much drone brood or no drone brood, we should see the opposite. So we should see a greater infestation of adult workers. This is actually data from 2 years. So I included the untreated controls from the first year that we ran and all the untreated controls from the second. So there's no treatment here at all. This is just what happened to the level of virus in the colony as the amount of drone brood changed over time. And we see that there's a strong relationship. So exactly as predicted, the more drone brood there is in the colony, the less virus there is in adult workers. However, you might argue the level of drone brood in a colony is actually correlated with a bunch of stuff, not everything that's captured here either. But at least one other thing that it's correlated with is how infested the bees are overall. So the overall Varroa infestation level, which we capture through alcohol wash, or not overall, but the dispersal phase infestation. And those things are related. So the level of drone brood is related to alcohol wash levels, and that's also related to the level of virus in the colony. Those things are intercorrelated, we call them. So what's really actually driving the effect? What's the important relationship? So we can do a fancy statistical thing where we remove the effect of one thing on the other. And when we do that, we find that if you remove the effect of the alcohol wash infestation rate from both of those things, you still end up with a strong negative correlation. So it's not the alcohol wash or the dispersal phase, the overall infestation level is not the explanation. It really does seem to be the case that it's the drone brood levels in the colony that are driving that effect. And again, we need to run some more tests. That's really it. The summary so far from what we've seen, the type of Varroa treatment actually affects outcomes in terms of viruses, and drone brood levels may actually drive the dynamics of how the virus is flowing through different populations in the hive and what's actually being targeted. So if anybody has any questions.
Could you just summarize, most of us are hobby beekeepers, with a small number of hives. What would you summarize as the key things that we can take out of that?
The key things is the type of Varroa treatment that you use actually might matter. So there's that. And the timing of your Varroa treatment might really matter too. I kind of didn't really get into that, but if you consider the arc of how the drone brood is rising and lowering, and especially if you consider that one of the main effects of the viruses that the Varroa transmits is to weaken colonies over winter, you really want to make sure that you are treating for Varroa mites before all of those Varroa mites transition from their drone brood hosts into the worker brood. So that's something that I didn't really talk about. And then similarly, you want to be treating early on in the spring rather than waiting until, later on in the summer when, again, Varroa levels are building up. And as I showed, it's not like they're not going into worker brood. They are going into worker brood. They prefer drone brood, but there's still some overlap into the worker brood. So if you can prevent that from happening by treating earlier on. So I would say for a hobbyist, to my mind, the key takeaways, make sure that you're treating at the very beginning of the season. Make sure that you're treating before you go into winter. If you need to treat it in the summer, in addition to that, that's fine. But as long as you've got those 2 time points covered, I think that you need to be treating them.
Okay, thanks, Heather, for your answer, and thanks, Nicola. Charles, you've got a question or comment, please?
Thanks very much, Heather. That was really fascinating. I'd like to say that the photographs of the Varroa and on the drones and in the cells was just superb. The other thing which I found really, really important for me was you've given me an insight into how the Varroa mites maintain their genetic diversity, because everything I've read up to this point in time, you have a sort of a foundress Varroa mite, and she has babies that mate with one another, but a species can't survive without any genetic diversity. And your observations that there are multiple female adult Varroa mites in the cells producing babies, and they must all mate with one another to maintain the genetic diversity. And that's the first time that I've heard anyone speak about that. And I think that's a really crucial point to our understanding of the Varroa mite. Thank you.
I actually wanted to speak to that a little bit briefly because my background is evolutionary biology. So that's something that I'm really interested in anyway. And if you want to read more on that, Charles, or anyone else, I actually write a blog for Nod sometimes. I haven't for a while, but I wrote a whole one on inbreeding in the Varroa mite. And there's actually way more going on than even that, than multiple foundresses. There's actually a bunch of things happening. So if you go to nodglobal.com, and there's a section for blogs. I think I wrote it about 2 years ago, and it's called Know Thy Enemy Part 2 or something like that. And it's like inbreeding in the Varroa mite.
Drew, you've got a comment or question, please.
I saw that you did the 1+1 and the 2 strips. Any thoughts about doing 1+1+1, which is actually what I've been doing for several years now, and it works really good. And I don't want to change it up because I have such success with it.
So do you do like one strip for 10 days and then one strip for 10 days and then one strip for 10? So 30 days total?
Yes.
Cool. And you're doing that like spring, fall? Like when are you doing that?
So this year I did it Labor Day weekend. Immediately after I pulled all my honey supers and the temperatures were low enough, below 85 degrees. That's when I start doing it. When I can look at the forecast and see, okay, it's going to be below 85. So this year I thought it was a little late.
Did you ever do just the 2-1 strips, like the 1+1, or have you always done the 3?
I just started with the 3 when I first started doing it, and I've just stuck with it because it seems to work. I do the Formic Pro as soon as I can when temperature's allowed, and then I wait for the winter solstice, and then I do oxalic acid treatments. And I have screen bottom boards on all my hives. So as I'm doing my oxalic acid treatments, I will pull those boards out and count the mite drops. And I will usually treat 3 or 4 times until I don't have any more mites dropping down.
I mean, honestly, if it's working for you, I mean, I think you should keep doing it. I haven't really thought about 1+1+1 before. I guess it's always like with formic acid, as other people have mentioned, it can be hard on your bees. So my philosophy is always trying to limit that exposure if I possibly can. But if it's working for you, man, like carry on. That's what I say.
Yeah. And as I said, I always lose some queens, but at least I lose some queens in a controlled fashion. And usually it's the weaker ones. I don't lose the queens out of the strong colonies. This year I lost a queen out of a strong colony while I was doing the Formic Pro treatment, but that was because somebody rolled a tire down the hill and knocked the hive over. And I think they were so agitated that they just took the Formic Pro and bake themselves out.
Yeah, I mean, things can happen, absolutely. I do think that most of the time it's going to be your weak queens or your poorly mated queens. But sometimes it's just it's a random thing. You can't always predict what's going to happen. So, yeah.
Another thing I was going to say, I play kind of a cruel trick on the bees. I use always at least a box of drone frames for my honey supers on each one of the hives. And I find what this does is the bees don't tend to build any drone comb in the brood box because there's drone comb available. So what they always end up doing is they leave a nice sphere of open drone cells that they don't put any honey in, that they're waiting for the queen to go up and lay drone brood. But the queen can't get up past the excluder. So most of my colonies are very few drones in any of them.
Interesting.
And I've often thought about this from a breeding aspect for my queens and just propagating some good genetics, because I don't produce a lot of drones for mating purposes. But I think it really helps with my honey production because they're not caring for all that drone brood.
Yeah, the colony doesn't have to devote any energy to it. You're right. Yeah. Well, and then in terms of like what I was talking about, the drones offering protection, I keep thinking about all these kinds of questions because people say, well, maybe it makes sense then to be doing like drone brood removal or something like what you're doing and limiting the amount of drones. But I think in the case of what you're doing, especially if they really aren't producing drones, I'm not sure that that's beneficial in terms of Varroa, because now the Varroa only have one choice, and that's to infest all the worker brood. They can't infest the drone brood. So I don't know, but if it's working for you again, and And in terms of honey production, I can totally see that.
You raise a good question, though.
Okay, Drew, thanks for the question, and Heather for the response. And I'd like now to thank you, Heather, for your presentation. As you can see, it's raised a lot of interest, and even though it's pretty detailed, people will be able to look at the recording afterwards and study it in more detail.